So the last interaction between the side chain is the weakest, which is the hydrophobic interaction. And all that is remember the non-polar amino acids that had basically carbons and hydrogens in the side chain. So most proteins are in aqueous environments. So this causes the amino acid chains that have a lot of hydrophobic regions to fold up, so that they're quote unquote inside the protein, and that way they could exclude the water, while the more polar side chain amino acids will want to be in the aqueous environment. All right, so even though the reason why I put a star there is that even though hydrophobic interaction is the weakest, they are sometimes the most significant force in proteins, because there are so many of these nonpolar amino acids usually, and even though they're very, very weak, the cumulative effect can be greater in magnitude than, like, a single hydrogen bonding or a single ionated attraction. So you can't disregard this interaction, and you have to pay attention to the cumulative effect when it comes to thinking about why proteins fold a certain way. All right. So the last level is the quaternary level, and that is how different peptide chains, we call them subunits, interact to form an entire protein. So the drawing that you have here are two different types of proteins. This is a picture of hemoglobin. And the hemoglobin, as you know, is very, very important, and we'll digress a little bit and talk about this. The quaternary structure is that there are 4 units of this. So if you look at each individual unit, it looks like this. So this is a chain, and it forms a globular shape, and 4 of these comes together and to make one molecule of hemoglobin. And that is that belongs to a type of proteins called globular proteins. And the example of globular proteins are insulin and hemoglobin. And they tend to be soluble in water. And they tend to have alpha helix and beta sheets present in these. And mostly their function is a metabolic. So they act as catalysts, regulator, or for transport. And that's what the hemoglobin does, is it transports oxygen for us. So that's the they tend to be functional proteins. And then the other type of proteins are fibrous proteins, as you see in this example. So this is the alpha helix, one chain, and then another chain, and another chain, 3 chains kind of twisting together to make a helical polypeptide chain. So that would be a quaternary structure, how these 3 chains come together, and they make up fibrous proteins, and these are found in keratins and collagens. And keratins is what's present in hair, feather, nails, and shells, and things like that. Collagen is what is present in skin, ligaments, tendons, bones, and teeth. All right, so they tend to be insoluble in water, and they consist mostly of alpha helix. All right, and these type of proteins are for structural functions. And like our skin is for to protect. All right, so that sort of concludes the 4 different levels of proteins. And what I want to do is briefly digress a little bit and talk about sickle cell anemia. And in real lecture, what I usually do is use this website, which is at UMass, www.umass.edu/microbiome/chem. This is a great website. You should look at the hemoglobin on this. Everything is in 3D, but I don't know how to do this with the document camera to record it. So I'm just gonna do it by paper format. And so I'm just gonna describe hemoglobin, normal hemoglobin first, and then we'll look at how one change in the amino acid sequence changes a life of a person. All right. So hemoglobin, the primary structure, and the reason why I like to do this is it sort of reviews the 4 levels, and it really looks at the function of how these things work. All right, so the primary structure of hemoglobin is, primary structure of hemoglobin is 146 amino acids, and the sequence of that is shown in this long. There's 146 starting from valine, that's the N terminus, and it keeps going and going. Wherever you see the minuses and plus are the acidic and the basic amino acids, and it keeps going. And this right here is the group heme, and that's sort of embedded in there, and the iron is what's responsible for carrying the oxygen. All right, so the interesting that you see here is that the iron is sort of chelated with the histidine amino acid because it's got the nitrogen lone pair to contribute to the iron. That's where it's held in space. So it keeps going, and the last one is the carboxyl end. All right, so there's 146 amino acids in this, and they're mostly alpha helix, as you see in this diagram, and that's the heme group. A lot of alpha helixes, and there are 4 of these subunits. Okay, so there is there's alpha unit and a beta unit. And what happens in one molecule hemoglobin is that 2 alpha units and 2 beta units come together to form one molecule of hemoglobin, and that is shown in this here. So you see in 4 different colors the 4 subunits, and together they form this globular protein. So there's 4 heme group for one molecule of hemoglobin. That's where the oxygen is carried. All right. So how is this different in a normal person versus the sickle cell anemia patient? So in sickle cell anemia patient, what happens is that there are substitutions in the amino acid. Instead of the normal amino acids that are present, there are other amino acids that are present instead of it. All right, so the one that really makes a big difference in terms of health is the one in which the amino acid at position 6, which in a normal person is glutamic acid, so it's negatively charged. That is replaced in a sickle cell anemia patient. So amino acid number 6 is glutamic acid in normal person. But if it's switched to valine at number 6, that's when it causes the most damage in a sickle cell anemia, right? So usually one substitution out of 146 amino acids doesn't impact health, but in this case it does make a difference because there's a big difference in the function of glutamic versus valine. All right, glutamic, as you see, is negatively charged side chain. When you substitute that with valine, it's a hydrophobic amino acid. So what happens is that a molecule of hemoglobin, which I'm going to draw as a circle, it's got this valine on the surface, which used to be glutamic acid, so that was no problem because it's in an aqueous environment. But think of this little spot that I've drawn as being hydrophobic now. That causes it to be sticky. So then it likes to be together with another molecule with the sticky part, on and on and on. So and you have these in 2 different spots, right? Because there are 4 units like that, and actually the substitution happens on the 2 of the units. So then it forms this long chain of hemoglobin rather than be globular and kind of free in space. All right, so they kind of form this chain like hemoglobin molecules. All right, so then what happens is it turns out that the regular blood vessel looks like this, and the width of that is 7 microns, and that's not a problem because it sort of squishes through the blood vessels because they're more malleable. It turns out the smallest blood vessels is only 3 microns in width. So a regular blood vessel in which the hemoglobin molecules are just floating around is able to squish through the 3 micron blood vessel. But what happens is in a sickle cell anemia, their red blood cells look like a sickle cell rather than a round pliable, and it's more rigid. So what happens is if the opening of the blood vessels like this and is small, and the sickle cell, if it's oriented right and it goes through, then it's fine. But if it blocks the blood vessel because it's long and not flexible, then it causes problem in that the tissue cells beyond this are not getting the oxygen delivered, and that's when they have the attack. Okay, so if you can see the electron micrograph picture of the regular blood vessels, blood cells versus the sickle cell blood vessels, and that's why this one amino acid substitution causes so much problem due to this hydrophobic interaction of the hemoglobin molecules sticking together and causing this shape, which in turn causes it to get stuck in the blood vessels. All right, so I'm going to end this right here, and I'm going to have a very, very brief lecture to finish this up.